So I found a very powerful quote. It's from, he's an American veterinarian who's worked at Tufts University, basically in the genetics field his whole life. His name is Gerald Bell. And he wrote a very good statement. He said, genetic tests are powerful tools whose use can have a significant positive or negative impact on a breed's gene pool. As with all powerful tools, they should come with an instruction manual on safety and their proper use. So kind of think of today's talk as being that. It's the starting us off on the road to having an instruction manual, how to use this and how to try and have positive impacts on our breed's gene pool and not make negative decisions.
Hold on, let's see.
All right, I'll just go ahead then, and I'll go to the third slide, which is the content slide. So I've got 6 areas I want to cover. One is the basics of genetic testing. Secondly is a history of color in our breed. Then we'll go into the detail of the color tests available, what the gene being tested is in each case. So color testing really allows us to venture safely, but I would say with an abundance of caution to broaden our gene pool. I'll then go on to health testing with genetic tests and the genes there that we're testing. And then the final topic is a look at coefficient of inbreeding, which can be calculated by pedigree, but now we can also calculate it by genetic testing, and then a summary of what we saw. Moving on to the 4th slide, the basic of genetic testing. So starting off with our sample, we'll take a sample of either cheek cells or a blood sample or a semen sample, and they're collected from the individual, identified, and sent into a genetic testing lab. The lab extracts the DNA, and remember, using a method that is specific to the sample. So if they say send in cheek swabs, you can't send in semen samples and expect it to work. After they've extracted the DNA, they increase the quantity of DNA, and then they place tiny samples of that DNA with reagents on these multi-well plates. Now, each cell on a plate runs a separate test. So one way of doing this is having these big multiplex testing SNP chip plates. They can run thousands of tests on each plate. So that testing is universal. It tends to have all of the traits that are currently known in dogs on a plate. And so your dog is tested for stuff that is irrelevant to a Great Dane at hundreds of sites. The companies Embark and Optimal Selection are examples of that testing methodology. Some labs you can order just a few tests on your dog. When the lab does this, they just use a section of a SNP chip plate, and you have customized the testing you've requested. So the DNA going into the well and the reagents going into that well are specific to what you've ordered. So this special or custom testing, it involves a little bit of human intervention. So it can increase the cost. But this testing can also allow us to do things like measuring the base pair number. That's very important in the Merle test. And when the Irish spotting test comes online, it'll be very important there. And when we get the Brindle test up to commercial standards, which is a future project that the Great Dane Club of America would like to participate in. So this entire process is normally fully automated using robotics and barcodes at all stages. unless you've ordered customized testing. So that's why, for 120 or 140 pounds, whatever it is, optimal selection or Embark can give you such a huge amount of information, whereas when you go to a lab where you're just custom requesting, like the UC Davis lab, the good thing is they can give you a bit more detail by doing this custom testing, but the difficulty is the difference in pricing. So on to color history, slide 5, if you're following along on a PowerPoint. Our breed has lots of genes that are interacting, giving us our beautiful breed colors. And for safety, we have mostly interbred in 3 color families, namely the fawns and brindles, the blues and blacks, and then the harlequins, mantle, merles. And these family groupings really ensured that the colors could breed true for the most part. However, it somewhat restricts gene flow across the color families, which can have a negative impact on inbreeding. And particularly, we could see inbreeding increasing in less popular colors. There are color genes segregating in the Great Dane which are undesirable and untypical of the breed. Tan points, chocolate, maskless fawns and brindles, piebald, and ticking. All but the ticking are recessive. Ticking is expressed on harlequins and merles, but not in the other colors. Blacks, blues, brindles, and fawns often have dominant ticking, but it's not expressed on their code. The mantle, which is also known as Irish marking, is desired for the spectacular effect it has on the members of the harlequin, mantle, and merle family. However, outcrossing to solid colors such as blacks, blues, brindles, and fawns results in the recessive Irish marking being covered up by a dominant solid marking. So, some color history. On slide 7, I compare a mantle merle to a solid merle, and I show you what the genetic difference is. Both are big M, small m, so they're typical normal heterozygous merles. But the one with the Irish patterning, the mantle merle, has 2 copies of the recessive SI allele. And then the other, the solid merle, you can see that there are markings now on the neck, on the front, down the legs. And this has either 1 copy of the solid or 2 copies of the solid allele. Right now, the UC Davis lab is conducting work being led by our own Marianne Zanetos. And if you have a mantle or another dog of interesting color markings, the Great Dane Club of America will actually sponsor, provided Marianne Zanetos allows the dog into the study. The Great Dane Club of America will pay to have the dog's Irish markings base pair count basically run. So any dogs you have with really interesting markings in the variation on this this mantle theme, we'd very much like it included in the study, and the contact person is Marianne Zanetos. So the first gene we're going to move on to, so color testing genes on slide 8, agouti, or known as the A locus. Technically, it's called agouti signaling protein. And actually, the gene has been known that it was out there for years. So we stuck agouti onto this signal peptide when it was discovered. The gene is located on canine chromosome 24. And in Great Danes, we see two forms. We see the normal AY, which means that depending on what's happening at dominant black, we either have fawns and brindles, or if dominant black is present, then we have normal black or harlequin, merle mantle. The bad actor in here is that AT allele. So it stands for 10 points, as you would see in a Doberman or a Rottweiler. And that AT gene is not super rare in the breed, so we do need to be cautious. Most Great Danes carry 2 copies of AY and they're fine, but the reason we recommend color testing at agouti is to avoid mating 2 AT carriers. It doesn't mean that you wouldn't breed from an AT carrier. You would just find AY individuals to breed to it. On to slide number 9. The next gene we're going to go to is brown or B locus. That's technically the tyrosinase related protein. This one's located on chromosome 11, and 2 copies of this gene segregate in Great Danes. The dominant big B gives us normal black pigmentation, and the recessive small b only expresses brown or people call it chocolate pigmentation a lot. Almost all Great Danes are carrying 2 copies of the big B, so they're BB, big B genotype. But color testing is recommended so that you avoid mating 2 carriers together. And you'll start to see a theme here. We're not necessarily about throwing out these alleles, these bad characters, as I call them, but we are about ensuring that we don't end up producing undesirable colors. Color testing at the D, or the so-called dilution locus. The gene here is what's called a melanophelin gene. And it's located on chromosome 25. And there are 2 forms that segregate in Great Danes. The big D is for full pigmentation, and the recessive D, small d, which causes the dilution of black to blue. But even in fawns or brindles, you will see that the fawn area of the coat is lighter as well. And it's because the small d effect, when there are 2 copies, is that it doesn't allow as much pigment to be placed within the hair. So you get a lot more light passing through it, and that's why a black now looks blue, or a fawn looks more champagne with a blue mask. The DD genes are widely segregating in the dogs. All blue Great Danes have at least 1 copy of dominant black and are homozygous for the recessive DD genotype. So color testing at the D locus is recommended to ensure that blue dogs are produced where desired, but avoiding producing blue brindles and blue fawns. On to slide 11. This is color testing at the extension locus, or E locus. This is one of the first color genes that was actually mapped, and we understood that it was the melanocortin 1 receptor gene. It's located on chromosome 5. In Great Danes, we have 2 copies. We have the masking, which is dominant, the E, capital E, superscript M as it's known. And this produces our masking, which is most seen. You could actually see the phenotype in our fawns and brindles. The recessive E results in no masks in brindle and fawn. And brindles and fawns with no masks are actually EE genotype. So they have 2 copies of the recessive E allele. Great Danes actually widely segregate. Many Great Danes from the black, blue, harlequin, mantle, merle families are in fact either EE, small e, or they are carrying 1 copy of the masking gene. So that's why very often you have to be careful when you're going to a black with your fawns or brindles, that you're careful that the black carries masking, so you don't end up with any maskless dogs. So it's certainly therefore recommended for testing in brindles and fawns to prevent maskless progeny. Color gene testing the harlequin or H locus. Technically, this gene is known as a protosome B7. The B7 actually refers to it's a loop that changes how the protosome gene would normally work. It is on chromosome 9, and there are 2 forms in Great Danes. So harlequin is dominant, and in the presence of a merle causes the actual harlequin phenotype. In homozygous merles, it causes the dogs that are almost all white. The recessive H has no effect on coat color. Great Danes widely segregate for harlequin. Many fawns and brindles actually carry a harlequin gene too. And the homozygous dominant HH genotype is an in utero lethal. That's because it completely incapacitates the proteasome gene from functioning. So color testing is recommended particularly in mantles. Onto slide lucky 13, color gene testing at the K locus. This is technically now called the canine beta defensin 103 gene. It's located on the chromosome 16. And in Great Danes, we have 3 forms of it. And we do actually know that the 3 forms are here. But what we don't have is a commercial test currently for brindle. So big K superscript B is what we call our dominant black. Big K superscript BR is our brindle, and this is dominant to fawn, which is small k superscript Y. And both the brindle and the fawn are recessive to black. Great Danes are widely segregating for all 3 alleles. So a black can carry either fawn or brindle, but not both. So a black that is carrying brindle is a big K, big B, big KBR in genotype, and a black that carries for fawn is also big K, big B superscript with a small KY. So color testing is recommended at this K locus pretty much across the board in the breed. Color gene testing merle. This gene, the pre-melanosome gene, so it is actually related to the gene that was functioning in harlequin as well. So this one, it used to be known as silver, and now it's actually the new nomenclature has changed to reflect the actual name of the protein being produced. This is located on chromosome 10. And as we know, there are 2 forms in the Great Dane. And I'm going to keep it simple here. So we'll just talk about M, the dominant, which results in merle. Obviously, we all know that there are base pair length variations in merle, but I covered that the last time we had a webinar. A merlequin is a homozygous merle, so MM, but it does not carry the harlequin gene. And recessive small m has no effect on coat color. So both the pre-melanosome and the melanosome genes, the mutations, which are both dominant, affect pigment being produced and placed in the hair. So Great Danes widely segregate for this gene. As I said, we have variation in what's called the A tail of the gene, and that's why it is important to go get that testing done at a place where they're going to give you the base pair number. So color testing is definitely recommended at this gene for all Harlequin mantle merles. And a black bitch with little bits of white that look odd. So Harlequins, as I said to you before, cannot be bred without the merle. So a Harlequin with this genotype, big H, little h, big M, little m, and this is our phenotype, the picture of the Harlequin on slide 15. The only difference between the merle and the Harlequin, as you can see in that genotype, is when there's no Harlequin there, we just have the simple merle pattern. All right, slide 16. This is looking at the solid or S locus. Technically, this is known as the microphthalmia associated transcription factor. It's located on chromosome 20. There are three forms of this gene in Great Danes. Big S, which is the dominant solid. Small s superscript I, which is our mantle or Irish spotting. And this is the one that we have a project right now where the Great Dane Club of America is sponsoring dogs of these colors to come in and be tested at the lab at UC Davis. And you do have to supply front and side photographs of the dog so that we can correlate genotype with phenotype. And there is also another recessive allele here, another one of our bad actors. It's the piebald. So our mantle genotypes can be the desirable SI, but we also see plenty of mantles that are carrying piebald. They still look like mantles, because only when you have two copies of the piebald, small s superscript p allele, do we see the actual piebald genotype. So Great Danes are segregating for all three, and only the solid S and the piebald alleles are commercially available. But as I said, the GDCA is currently funding a study to validate the SI test with UC Davis Veterinary Medical Genetics Lab. And the last gene, it has not been sequenced. This is ticking. So we don't know what chromosome it's located on. We do know that there are two forms of ticking segregating in Great Danes. Big T, which is the symbol for dominant ticking, and small t is recessive and results in clean white areas in Harlequins. Great Danes widely segregate for this. Most Harlequin mantle and merles have been cleared of ticking, so they would be small t genotype. There's no commercial test developed for this since the gene hasn't been sequenced. Outcrossing of Harlequin mantle merle to solid colors such as black, blue, brindle, and fawn risk reintroduction of the ticking gene, because the ticking gene, while not expressed on the black, blue, brindle, and fawn pattern, can very often be present. Okay, switching topics now on slide 18, genetic health testing. First of all, I list the two known health disorders of Great Danes with a genetic test. Central neural myopathy. This is caused by a mutation in the bridging integrator gene, BIN1 gene. I love that name. and leads to a rapid progressing muscle myopathy. Less than 20% of affected Great Danes reach adulthood. Affected individuals are homozygous for the recessive mutation, which was discovered in Great Danes on chromosome 2. The other condition is ichthyosis. This is caused by a mutation in the splice acceptor site. And it leads to epidermal, meaning skin, and follicular, meaning the growth sites inside the skin for hair. They become either orthokeratosis or hyperkeratosis. Those are two terms for two forms of cornification. One is where there's bony type hardening within the dermis or the skin, and the other is where the skin overproduces it's tough and inelastic tissue. That's what those two terms mean. The cornification tends to be concentrated in the skin of the head and the extremities like the feet. Affected individuals are homozygous recessive for the mutation. And again, this was discovered in Great Danes, and it's located also on chromosome 9. Now, on to genetic testing health disorders which may help Great Danes. It is believed that von Willebrand's disease type 1 is present in Great Danes, and that the causative mutation is in the von Willebrand's factor gene. It causes a defective blood clotting protein to be made. Affected individuals are homozygous recessive for the mutation, and the mutation, the gene is located on chromosome 27. The other condition which does occur in Great Danes is multi-drug resistance 1. This is actually a mutation in the transporter P or polyglycoprotein. Affected individuals that are homozygous recessive for the mutation located on chromosome 14, it affects their ability to metabolize some common drugs like ivermectin, loperamide, and lots of other really common drugs. So the dogs that are homozygous for this really have a very compromised ability to take advantage of a lot of drugs. Okay, slide 20, we'll start the topic of coefficient of inbreeding, COI, calculated by pedigree or calculated by genetic testing. Let's start with the calculation by pedigree. This was developed by a British geneticist, Sewell Wright. Sewell actually trained my academic grandfather. And what Sewell's work did was he developed a way to calculate the probability of two alleles that are identical by descent from a common ancestor. So the more ancestors shared by the sire and the dam, and the more recently these ancestors appear in the pedigree, the higher the COI is going to be, because the likelihood of genes coming from the same ancestor, the more frequently the ancestor appears and the more recently it appears, the more it'll drive up the coefficient of inbreeding. This method, while easily calculated, has a few limitations. In order to make these calculations manageable, people usually just use about 10 generations of pedigree data. And that's using a computer to do the calculation. That's not, if you were to hand compute a 10-generation pedigree, that would be a big chunk of time out of your life. What this method doesn't do is it doesn't allow for mutations. Well, most mutations have no significance whatsoever. A small handful are either negative or positive for the animal's chances of being selected for breeding. A few other things about Sewell-Wright's coefficient of inbreeding. It assumes that all ancestors have equal merit. They are not. Some animals are really superior in terms of their ability to pass along genes which come close to the breed ideal than other individuals. So therefore, they're not all equal. Each individual has a unique genetic load. Genetic load is the number of deleterious traits carried by that individual. If we confine ourselves to looking only at recessive traits, an average individual carries 3 to 6 different deleterious recessive alleles. Now that doesn't mean, so that means it's got a copy of the normal form of the gene, so the individual is perfectly normal looking. But it does carry all these recessives, which if they become together in their progeny in the future, would result in the expression of that deleterious trait. So an ancestor who was not only genetically superior for the desirable traits, but is also desirable because of a low genetic load, will still get the same calculation of coefficient of inbreeding than we would get with an extremely high genetic load individual. So what I'm saying here is, if you're line breeding to a dog that was transmitting good genetic type for the breed, and the individual had a low genetic load, then being inbred on that individual doesn't seem very undesirable. Examples in the history of our breed would have been Bosco and Dolph in Germany, and you can go down through history and see where there were some really great dogs that were foundational to our breed, and so what I'm saying is, we're not talking about inbreeding always being bad. We're just saying that it's a very nuanced situation. So it's very important that you decide to whom you want to line breed. Okay, on slide 22, let's talk about the calculating by genetic testing now of the coefficient of inbreeding. So how the testing does it is it measures how much of the genome tested is homozygous in an individual. In other words, how many areas in the DNA are we getting the same allele at both sites on the chromosome. And so we can calculate that again as a percentage that are homozygous for the same allele. So it actually ends up with a very similar number to what the Wright's coefficient of inbreeding would be. But what it will allow us to do in the future is, since we will know what are desirable alleles and what are undesirable alleles to have at various different loci, then we can look at the percentage of deleterious homozygosity in an individual. So how this looks is, from a recent ancestor in common, you'll see on a chromosome, you'll see a whole area that is in common. So you'll see these long parts of the chromosome that are just identical as they're descended from that individual. So an ancestor who is in common and was itself highly inbred will result in a much higher percentage which is homozygous for the same allele. Again, we don't know if high COI was to an individual of high or low genetic load, and that's a very important point. So on to the first summary slide. Genetic testing has given us almost a complete picture of coat color genetics in Great Danes, but let's be sensible. and cross-color families only with great care and with due planning for future generations. Remember Nellie Ennals about saved blue Great Danes from extinction post-World War II. And if you doubt me making that statement, run back any pedigrees of blues today to their 1940s ancestors and you'll see just how important her work was. She did this by clever mixed color breeding. Well, we have a handful of health conditions we can genetically test for today. All of these conditions are rare in the breed. And genetic testing for more common challenges, such as dilated cardiomyopathy, wobblers, megaesophagus, are in various stages of research, and we do hope that those will eventually give us a genetic testing for those traits. We actually have a very large healthy gene pool across the globe, so I encourage breeders to cooperate with those other breeders who do traditional health testing, publish their results, as well as using the new tools of genetic testing. Slide 24. Carriers of recessive traits, even undesirable traits, if otherwise worthy of reproduction, must be used to maintain a broad, healthy gene pool. Breeders with a non-carrier for trait X, but still only want to breed to other non-carriers, are going to rapidly narrow the gene pool. This is dangerous behavior. It's a case of a little information and not coming to the right conclusion. Once you get comfortable with all the new scientific mumbo jumbo around genetic testing, you will find it a brilliant tool to help you not kind of sweat that small stuff. Because a well-balanced breeding program gathers all the information possible, but one must prioritize. Make sure you go from the big picture and then go down into the smaller details. So I thank you again for giving me the opportunity to present this to you, and I will open it to questions.
Yeah, I'm just asking everyone to unmute, Neil. Okay.
Great. Oh, now I can see. Sorry, I was reading off the. I was reading off my copy of the PowerPoint, so now I see you got it up and running on yours.
Yeah.
So, any questions?
So just ask.
Please.
Neil, can you, Neil Secretary, can you take off the PowerPoint now so that we can see everybody? Ah, that's
There we go.
okay. Thank you. That was interesting, Neil. It's quite complicated for somebody like me, but I think I was starting to understand it a little bit.
Right. I think the way I've structured the PowerPoint, hopefully, it will, end up as being a resource long term, where you've got a particular question, you go, oh, let's go back, look up that gene, let's see what's happening. Or when people are thinking about inbreeding, I want them to think about the picture, the big picture. Because there is a tendency, and for example, the rules that the Swedish Kennel Club brought in about inbreeding, well, they really don't affect Great Danes very much because we don't tend to inbreed very tightly. In breeds with a very small gene pool, you almost couldn't breed your dogs and follow the rules in Sweden. And of course, I'm very familiar with the Irish soft-coated Wheaten Terriers, and that would be an example. But many of the British Terrier breeds would really struggle to meet those requirements. Skies, Sealyham Terriers, Welsh Terriers, even Lakeys.
you on mute there, Sandy?
I'm on mute because I've got a shouting dog in the background. They just had a thing, so they're shouting. So I thought I didn't want to disrupt everything. So I'll stay on mute until I've got something to say.
Has anybody who's in the meeting today, have you sent any samples for our study on mantle Irish spotting, the validation study at UC Davis?
All of mine have been done.
Thank you. Thank you.
I
Thank
have,
you.
Neil.
Great. And so far, you know, the data seem to be suggesting that we'll be able to have a good solid recommendation for people moving forward. You know, for everybody in the Harl Mantle Merle family, it really is important that we keep up our color testing, because we can then kind of not be sweating that small stuff when we know exactly what we have color-wise. Shopping for the right mates gets easy.
Neil, I think I made you aware that the Great Dane clubs of the UK are putting out a questionnaire to all their members regarding the Merle, and so we're waiting for that to finish, which is around about the 22nd of May, and then that will be fed back to the Breed Council, and depending on that decision, we'll take it to the Kennel Club to see what their thoughts are.
It was a very pleasant surprise for us when you know, we obviously had the Harlequin Mantle Merle people, breeders, pushing the Great Dane Club of America, you know, to allow the Merle, and I was one of those very vocal people. But when we put it to the membership, you know, do you want us to investigate this? We got an overwhelming response, a positive response. So even people who have spent their whole lives in fawn and brindle world really care that the Harlequin people have the tools they need to breed well.
Yeah.
And gosh, I don't know if you've seen it, but we have some spectacular Merles in the ring in the US at the moment. You know, and you think about all the ones you never saw. A shame.
It was so exciting being at the Great Dane Club of America. What was it? 2000.
That was in 18. That was in 18 when we did the special
Kansas,
presentation.
wasn't it?
Merle, yeah, and Tracy Powell brought that gorgeous young Merle bitch, and what could you say? You know,
I picture her now. She absolutely the most beautiful shape.
We loved her, didn't we, Sandy?
She was so pretty, wasn't she?
Yeah.
oh, and she had the honor of being the first Merle to finish her championship in the U.S. as well, which was a super icing on it. Of course, you know, she took the bitch to breeder judges, so you know she didn't mess around with all rounders, which is a shame with our breed. You know, our breed judges are so good, and the people. I don't know, we do a terrible job educating people who don't come from our breed to judge our breed.
Neil, can I ask you a question? As far as a breed standard for the Merle, how difficult was it for you to get one out there?
So what we actually wanted to do was we had two things. One was we had, again, all-rounder judges obsessing about pattern and markings in our dogs, whether it be heavily marked brindles or lightly marked brindles, or how much, you know, how much black was on a Harlequin, all this kind of thing. So we restructured the way the standard goes through the colors. So we were very explicit. These are the 6 accepted colors. And there was a description. And then we talked about markings and pattern. And then at the end, we had a line which what we stated was the important thing to focus on in our breed is structure and type. You know, variation in color and markings is of no significance when it comes to structure or type. And so we wanted to do away with, you know, people fussing about a fawn that had, you know, a bit of black on the front, a bit of smudging, or fussing about whether a brindle was too dark or too light, or, you know, how much white was on a black. Because I can tell you, you know, blacks or blues with the same amount of white on them as a brindle or a fawn get penalized differently.
Yep, absolutely.
So we took the opportunity to really tell people, de-emphasize this little minutia of pattern and markings and concentrate on conformation. If a dog is a Harlequin, it fits this description. There's no two ways about it. It's a Harlequin. That's all you need to know about that dog. Don't talk to me about where its spots are. It's none of your business. Get on with judging the dog. Is it typey? Is it sound? You know. And I do think our new standard, I've had judges come up to me because people said, who came up with the phrase about emphasizing, and really it was from things Ray Cataldi and other people had been saying to me over the years. So it just like, it flew off the pen. And when the rest of the committee saw it, they were like, that's it. That's what we want to say in our standard. You know, concentrate on conformation and breed type.
Yep. Thank you.
A blue dog is a blue dog is a blue dog. Now, is it a well-structured blue dog? Is it a typey blue dog? This is what matters, not the fact that it's a blue dog or if it has a bit of white on it.
Neil, going back to the colour testing, what do you suggest someone with a Harlequin, what they would test for?
Yeah, so actually with Harlequins, so in the UK, I might give you a break on dilution because you're allowed your blue Harlequins. But I would literally go and run every single test on members of the Harlequin family.
Okay.
Meaning I'm going to test in agouti because I don't want to produce anything with 10 points. I'm going to test at B because I don't want any chocolate Harlequins. I'm going to test at D because I want to know if I would potentially get blue Harlequins, which for me, my end of the world would be a no-no. Extension, yeah, that doesn't really matter to Harlequin breeders. And then obviously we're going to test for dominant black because we want to know if these dogs carry the yellow recessive. So again, we're trying to prevent either fawn Harlequins, or merle or brindle Harlequins. And then I'm going to test the H gene, obviously, unless I actually have a Harlequin and I know, because if you have a Harlequin in front of you, you know the Harlequin genotype. I'm going to test the merle gene, and I'm going to test the solid markings to avoid producing piebalds.
Fantastic.
And you know, there is discussion, as you know, among the European clubs and led by the German club about is the FCI standard worded too loosely and basically allowing piebald to slip in. And so they're looking at probably cleaning up that language when they recognize the merle, which obviously COVID completely gummed up that works. You know, in 2019, as you know, we were making great progress on that. But since then things have slowed up.
Okay, right. Carry on from that. What would you test a mantle for? I'm asking these questions because there are quite a few people in the UK that still don't colour test. I do. So I know what I colour test for, but this will give people an idea of what they should be testing for if they had a harlequin, a merle, a mantle.
Right. I actually do want to have the information on the merle gene, even if I'm testing a mantle, because as I said, our cryptic mantles, cryptic merles are often coming under the cloak of a mantle, and so we need to be careful. So again, I'm talking about testing at A locus, B locus, D locus, K obviously, and then harlequin, merle, and solid. Same issues. You know, in one of the slides there, I showed you how, for example, a mantle could hide a piebald gene, as can a harlequin. And while it doesn't, we don't think at least it affects the pattern of that dog's phenotype, it will cause us to produce a mismarked dog in the future if we breed two piebald carriers together. Does that cover it?
Yeah.
Yes, that's good. So I assume that if you had a merle, you would do all the tests as well.
Yeah, except we can be pretty sure with a merle that it doesn't carry harlequin, because harlequin will always modify a merle. So you can drop your harlequin test on a merle, but test for everything else. Mm-hmm. And one of the things I wish so you know, the harlequin family of breeders really have to go and do some of that detailed testing, because you do want to know the base pair length of the merle genes you have, and in the future you know, in the future, I think we're going to have a simple answer for the actual mantle, the Irish spotting, but you know, all the data isn't in yet. So
fantastic, that's good. So, do you colour test your fawns and brindles?
I do. I most certainly do. Yeah. What I'm looking for there is, I'd love at some stage to have a really typey fawn or brindle male that I produce that carries for blue, because I would use it to help work on blue-black breeding. But mine tend not to, and I've outcrossed, you know, and my fawns and brindles tend never to carry any recessive dilution, whereas I know others have. You know, if you look at the Owl Watch dogs, they produce some of those gorgeous blues that they produced because they had an excellent fawn dog of their breeding that carried a blue recessive, a dilution recessive. So I'm a big proponent of it. I don't want to produce a chocolate. You know, my dogs go back to Mount Dania breeding and Giacomo breeding. I know the chocolate gene segregates down from those lines, so I want to avoid chocolate. I actually have had some in my line that have had recessive chocolate. The other thing that it's a big discussion, but not maybe for today, is many breeds have these really fantastic websites that are a database for their breed that are maintained by a parent club or a charitable organization. So, you know, most of you know I'm also a Wheaton person by marriage. But we have a fantastic database, the soft-coated Wheaten Terrier database. It has, you know, it's a small breed, but it has over 70,000 dogs on it. It's co-administered by an English breeder and an American breeder. So, you know, we have true international cooperation on it. But in there, you can run reports on, you know, what dogs have been tested, what they've been tested for. You can look at their pedigrees, but the amount of detail on health in this particular website is such that we find it very easy to recruit. So the National Institute of Health in America uses the soft-coated Wheaten Terrier and that database to study longevity. So we encourage everybody when their dog passes away, you know, to do whatever's necessary to get the veterinarian. You know, it isn't always necessary to necropsy, but sometimes you'll need necropsy, and you know, have then a definitive cause of death. If your dog had already been diagnosed with melanoma, for example, and eventually is euthanized because of that, you know, you have that diagnosis in there, and it makes it easier for researchers to work on new areas of interest. And we have a great pedigree database in the Quintessa database, but we don't have a link to the pedigrees to also the date of birth, health testing done, genetic testing done, date of death, cause of death. And it's something I'm planting the seeds in people's mind. This is something that would advance our breed a lot, I think, if we had access to that. Most of us publish all of our information on the Orthopedic Foundation of America website as well. But you know, they charge us to upload our information. So when I do genetic testing, for example, and you go look up my dog that is genetic testing that's on there, I had to pay to have it genetically tested, and I had to pay to have it put on the public website. So I think we need a way, you know, to do it in our breed and serve our breed. And it's got to be international. You know, it's got to have all of the main breeding programs around the world cooperating so that we can do better. There's no doubt the Great Dane has become much longer lived in my lifetime.
Yeah.
There's no doubt about it. The average lifespan of our dogs has increased. And I think that's come from better sharing of information. You know, I think you all know the story of me as a kid. You know, my first Great Dane, I got it from Jill West. Kilcrony Kilviscount was his name, and he was a brindle that, you know, we were bursting with excitement to start showing in the breed. And my father was very supportive. He knew he was losing to his Irish setters, but on I went. Well, you know, at 12 months, the Bruce, as we called him, started to develop wobblers, and got a hold of Jill, and Jill said, "Oh, you need to get him to the vet college." And she said, "This is definitely something in our breed." And she said, "You know, I think there's a genetic component to it." And I was only 14 at the time, and that made a big impression on me. And, you know, we had to euthanize him. At that stage, the surgery to stabilize the neck vertebrae hadn't been brought in. But it did, you know, her honesty as a breeder really shone a light to me. But it also, you know, I was fascinated. This is genetics. So, you know, I wanted to know more about genetics, and it gave me an entire career as well as a hobby. But I think that openness has gotten much better too. You know, people openly talk about what's going on in their breeding programs. When I hit a brick wall with my blacks and blues on dilated cardiomyopathy, you know, nobody was mad at me for sharing the information publicly. They were happy. you know, that plus, you know, they obviously knew that for me it was pretty devastating. I couldn't breed down from those dogs. I got to a stage where every male in the last couple of litters all died from DCM.
Wow.
Yeah.
Neil, how can we?
Yes.
What can we do as a club to contribute to the health testing and genetic health testing? And
I think to both encourage people to health test, both the genetic and the conventional health testing, but also we really need somewhere where it's on a public database, the information's on a public database, so that long term, you know, if I'm struggling with hip scores that are high, you know, I can start to look at families of dogs that have low hip scores, and so, you know, I can start selecting dogs with the characteristics I want and the health, you know, that'll help me improve, that kind of thing. But it really's got to be done in a public way. We need a public database. There was an initiative in the UK, and I don't know how it progressed, where there was more discussion about, you know, how to make progress on health issues in the breed. I think Tony Pierce was one of the people who was leading that up. Does anybody remember what I'm talking about?
Yeah, I remember what you're talking about.
I do.
And people at that time didn't want to share their information. So no one wanted it out there that their dog has got this or their dog's got that. So it was all forgotten about.
Right. Right.
And
but it is something that we're looking at again to do. So hopefully in the coming months we will discuss it and we will try and get a database out there. There's lots of breeds. The Spinoni has a brilliant website with all health testing. So any Spinoni wants to check the history, you click on, it's all there. So hopefully we will go down that road again.
I mean, we've already started. Sorry, we've already started with a health survey, and we've had, you know, a really good response to that. We've had today 871 responses. Sadly, some of the bigger breeders haven't taken part in that, which is a little bit disappointing. That's you know, but it's a good start. It's somewhere to work from.
Yeah.
Yeah, you know, I will tell you, as somebody who, you know, we've bred a lot, particularly in Wheatons, and for us just to keep all that information up to date in the Wheaton database, it's a lot of work for Jared and I. You know, and we're getting older, so I can understand why some of the bigger breeders, it becomes a daunting task getting all that in, but it's very satisfying once you get it in there. It really is, because you know that's your body of work. It's your lifetime body of work, and it is hard work to put it into a database, but trust me, it's so rewarding.
Yeah. Hopefully that's the road we'll go down next.
Well, I'm making my way through mine, filling up mine in at the moment. If you've only got about 4 dogs to do on that health thing, it's okay. But when you're looking back on like 30 and 40 dogs, it's like, where do I start with this?
It is. But Sheena, you wait until we've got to go through it all at the end. You know, I know it's difficult. I mean, I think I put about 15 dogs on there myself, you know, but you know, it's time consuming. But on the other hand, you know, when we come to reconcile all of this at the end, it'll be such a wealth of information that will help us.
Well, I'm working my way through it.
Thank you.
It's just taking some time, that's all.
I'll stop picking on you. Thank you.
Yeah, the problem has always been, I remember when Tony Pierce started that it was mainly to do with wobblers, wasn't it, Maria?
It was.
And nobody in the UK had ever bred a wobbler. Nobody. No matter who you asked, whatever, you know, nobody had that. They knew of someone who had.
Yeah.
They hadn't.
Well, this is it. It's getting people to be honest.
Yeah,
and out there. That's the only way we're going to get rid of it, is being honest. Like when I had the inherited myopathy, I was mortified. I put it out there that I had it. Then went down the road that we found out the labs had done it, published it. You know, I was at every show giving out information. Get your dogs tested. Get your dogs tested. Now, I had 2 in the litter affected, but I was out there saying I've got it. I didn't invent it. I didn't plan it, but I want to do something about it. And I think everyone is waking up now to the problems. They're not burying their head in the sand.
I hope so, because it still goes on. The secrecy and and it's on both sides. Breeders are secretive for a reason, and that is because pet owners and other breeders slaughter them if anything happens. You know, it's like, oh God, you did it on purpose. You actually went out there and bred a wobbler because you wanted to. So it's a two-sided thing. If the blame culture receded a little bit, then more breeders will be willing to come out and say, yep, okay. I remember I used a dog, very famous dog, and got a wobbler in the litter. And when I spoke to the owner of the sire, the first thing he said to me was, oh, I suppose you're going to tell everybody now, are you? That was he said, not, you know, nothing, just, I suppose you tell everybody now. Well, my dog's never produced a wobbler before. I said, well, funnily enough, neither is my bitch. That is the attitude that a lot of breeders have. Don't ask me if my dog's produced a wobbler, because I'm not going to tell you one way or the other. And, you know, it's seen as some sort of unclean thing that you've got to hide from everybody. And it breeds this culture of people not wanting other people to know and be honest, and actually further the breed because they won't be honest.
And it is so difficult, because without the information, you can't formulate plans to move forwards and to try and help the breed, can you?
No, you can't hope to even start finding tests and genetic problems.
I think just to cut in there, Sheena, I think one of the biggest problems about people being honest with everything is social media.
Oh God, yes, absolutely.
Which has taken it to another level. So people are not potentially being honest because they know that social media will take them apart, and that's the problem.
It's not just on things like that. Everybody's got an opinion on what you do if you breed a litter of Danes. Everybody.
And more importantly, while we've been having this seminar, one of my Danes has eaten all the sausages and it started snowing. I just went off. I heard one of the bowls crash in the kitchen, and I don't know who it was, but there's only one Dane there and she's denying it, and all the sausages have gone.
It was a dog that was walking past who broke in, and who knocked the bowl off and ate the sausages.
Yes. Yeah, and she's denying all responsibility.
And I just say I didn't breed that.
How do we know?
And it's still snowing.
Going back to the colour testing as well, recently I saw on one discussion thread on social media someone saying that, not them in particular, but the person that they get their dogs from doesn't need to colour test their Harlequins because they know their lines and have known their lines for years, so they know exactly what colours their dogs are going to produce. How do you get it over to people like that? That's just not the case.
I actually have a really smart-ass quip for that. I say the only people who seem to know all about their own dogs without doing any testing are backyard breeders.
Yeah.
You would faint if I said that, because the breeder in question is supposed to be the massive big authority on anything spots. But
Can I show?
I stand over my statement, Sheena, and I would say it in another seminar. If you're not testing in this day and age, you are nothing but a glorified backyard breeder.
Yeah.
I don't care how many champions you produce.
oh no, it's not got many of them. But
Can I show the picture of my black bitch so people can see? I originally kept her with the idea of putting her with a Harlequin just by going by her colouring. And it would just, if I can show it on here, then it'll make people sort of go, oh. So I don't know if that's
Yeah, I can see that.
yeah.
No.
Can't see it, Tammy.
You have to keep talking, Tammy, and then you come up big.
yeah, keep talking.
Oh,
Tammy, your face will come up.
That's
That's
it.
That's it.
Oh,
Keep
wow.
talking. La 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 la.
So and
She's got white blade, white coming down here and on the back.
But she's the cryptic merle, isn't she, Tammy?
she's a cryptic merle. She has not got a touch of merle on her.
Mm.
Wow.
So
Children,
she
it
cannot
is.
be bred.
I bet I could stalk out. I mean, other than, so the reason she doesn't have, she may have a little bit of merle, but her Harlequin gene, because remember she has a Harlequin gene,
Yeah.
as well as a cryptic merle gene. So that means that in some areas of her body, she's expressing like a Harlequin. So where more of the merle would have shown up, because she's got that Harlequin gene, it's really turning the merle to white.
Yeah. Yeah. See,
Absolutely fascinating example, though.
I don't even know why I ticked the merle gene box, but that's why it's made me go, holy shit, I've got to do it on the mantles and the blacks with white, so I know what's inside, because I can't see it.
Yep.
Yeah.
Absolutely.
It's actually
And the other great bit of information, you know, you got out of this is, you know, she's obviously a lovely bitch. You still want to breed her, but now you're going, ooh, I need to go mantle with her, not Harlequin, because I don't want white puppies or Merlequin puppies.
Yes.
Yeah.
it's quite
Absolutely.
an exciting thing, isn't it? The fact that we can now test for all sorts of different colours. I remember just talking about Nellie Ennals. I remember 100 years ago, at least back in the Victorian times, I did a brindle to fawn mating, and I got a blue brindle. And I ran screaming from the building because it was something like about my third litter. And I ran screaming from the building and wondering which person on this pedigree on both sides that I looked at had lied through their teeth, and where the blue was in this pedigree. I was going absolutely daft. And it
Beauty of Brington Hill was back there on both sides, right?
Right at the back, yeah. It went back to Klaus Denton and all the rest of it. But
yep.
I mean, Nellie, when I said to Nellie, just said to me, "Oh, calm down, woman. It's perfectly normal. It's not very common. Perfectly normal." And I was I couldn't believe my eyes. I could not believe my eyes. I was a fawn brindle breeder, and out had popped this blue brindle. I was just like, oh my God, what has come out of my bitch? And I thought all sorts of skullduggery had happened, when in actual fact it hadn't. You know, it was explainable.
Yep, it was that dilution recessive just hanging out there in the pedigrees forever. And there I am, Sheena, trying to find a good fawn dog with it.
Yeah, exactly.
And by the way, you know, this is an aside, but you'll notice that I think it's tricky to take brindle into black and blue breeding, whereas it's not tricky to take fawn, because some blacks that carry brindle will show a degree of so they'll have black stripes and they'll have more intense black stripes. And I'm not imagining it because, you know, years ago I discussed it with Carolyn Mobley, and she had brought a Mountainea dog out to California to improve their stuff. And she found that when she bred him into the brindle dog into the black lines, that about half the blacks were not showable. And it turned out when she'd breed them, those were the ones that had brindle in them. Whereas the fawn doesn't seem to be a problem.
Yeah.
But, you know, there aren't enough, since we don't have a test commercially available for the brindle yet, it's hard to prove what I'm saying till that test comes along.
What will you need, Neil, for testing the brindles?
Yeah, so what we're going to do is the same study design as we are doing right now with the mantle Irish spotting. We're going to get pictures of dogs, and the GDCA will help pay for the K locus testing, because again, it's just so the length of the gene for dominant black is longer than the length for the small k, small y. And we believe brindle is intermediate in length. And we know, you know, we know most of that. Now we need to start doing the testing on volumes to validate it. And it would be interesting to have, if we can find blacks that do carry brindle, to try and get good photographs of them to see if we see this striping on a black dog. I see Sheena's nodding. Has anybody else noticed what I'm talking about? On blacks that carry?
Yeah.
You mean a very, very dark brindle? What we call?
No. These are black dogs. They will produce black progeny for sure, but they carry for brindle. And unlike a black dog that carries for fawn, these blacks will have 2 shades of black on them. And you'll see the lighter shade of black is in that chevron type brindle pattern. And another person who was of that belief as well was, I'm sorry, but I always called her Mother Robery. And you know, she did a lot of fawn black breeding in the UK. And again, you know, she would never go to some of the old manor brindles because she was worried about that. And I remember when Jill West bred Captain, Mother Robery had sent her quite the letter telling her off for doing the breeding, because she bred to an old manor brindle dog to her black bitch to produce Captain. But Captain's color was fine, so.
What I'm picking up at, well, sorry,
I've just got one little question. I don't know whether, because obviously, I mean, I'm new to all this. I've never colour tested a dog before. I mean, the genetic tests I've had done are the Labocline, is it, I think? Do we, I mean, who do people use in the UK for the colour testing? Tammy, I assume you use a regular, is it Labocline you use, or?
No, I use UC Davis in California for my colour testing. They also do the same DNA genetic testing as what Labokin does.
Yeah.
I prefer the layout that UC Davies give you in the colour. Very easy to.
And it's easy enough to send your samples across and everything. That's not an issue.
Absolutely. No. I did some tests recently. It does work out sort of cheaper with the exchange rate, and to post my DNA samples over to America cost me £2.85. They got that within 10 days.
Brilliant.
Yeah.
Okay. Right, brilliant, thank you. Anybody else? So any colour
We've
testing
used Laboclin in the past, Greta.
Because you specifically have to say which colours you want doing with that one, don't you? Whereas, I think, am I right in saying, Neil, the Embark and the Optimal Selection, do they do across the board tests rather than you having to specify?
They do, they do, but for example, with their Merle test, they won't give you the base pair length of the A tail. So I guess what I should do is get another sample from the cryptic mantle bitch, the cryptic Merle mantle bitch that Mags has, and send it through Embark and see whether they pick up, that she actually has a Merle gene or not. All they do is they tell you, Merle and non-Merle on the Embark, and it's the same with Optimal Selection. So one of the things I've done is send the same dogs into a number of different labs. So I've done a little bit of quality control. Just because, I'm giving these seminars to quite a number of clubs and breeds, and I want to be able to, have skin in the game. If I say, I can recommend this, and like Tammy, I just love the way UC Davis in California do the color testing. Plus, with UC Davis, you can also check a box for the two specific diseases to Great Danes, as well as the multi-drug resistance and the von Willebrand type 1.
And do they do those tests at the same time, the UC Davis? Because I've obviously had various done already by the Laboclin or whatever it is, but obviously if I'm looking at colour testing as well, I've never tested a dog before, so this is something that I need to think about going forward, to use somebody who does everything, who's suitable for everything.
Yeah, see, my take on it is Embark and Optimal Selection seem very cheap because they give you, I don't know if you can all see this, but this is a report here on one dog, printed on two sides, okay? It's absolutely sheafs and sheafs of information, but it's mostly about things that occur in other breeds of dogs and not in Great Danes. And I'm just testing my Great Dane here. But what they do is they have a fixed multiplex plate, put a little drop of the dog's DNA into every single well, and it's automated from start to finish. That's why it's cheap. What UC Davis is doing or Laboclin are doing they're customizing part of a plate for your dog. And, in goes your dog's DNA plus the reagents to test it for Harlequin. In the next well, testing for Merle. In the next well, testing for Solid, and so forth. And that's the way they're doing it. So there's one step in that, or one stage in that, where I suspect they haven't automated it. You actually have a lab tech, pipetting there, and that's why the tests are a bit more expensive, because they're more labor intensive.
Yeah, because the ones I'm currently doing, I'm sort of doing my research, doing a test, doing a bit more research, doing another test. If there's obviously somebody out there that will say, do a Great Dane and I will do all of these tests, and then you can pick what you want to look at. For me, that would be probably a slight deal less expensive than what I'm currently doing now.
The thing is, Neil, on your on your presentation, you say about cooperate with breeders who do traditional health testing, as well as obviously new tools. But what are the traditional health tests that you would either you guys do over in America or that you would recommend to do?
What I want done, and what I do on my own dogs, is my Great Danes have hip X-rays, elbow X-rays. They have patella palpation for looking for subluxating patellas. Now, I've never found that in a Great Dane, but it occurs in Wheatons, and it doesn't take very long to do it. Thyroid testing, and the thyroid panel we do is one that's actually approved by the College of Veterinary. I'm blanking out on the word beginning with E. Anyway, not epidemiology, endocrinology. Thank you. The old brain works still. So I have thyroid testing, and specifically what I'm looking for there is if there's the presence of anti-thyroid antibody. We do a complete Doppler echocardiogram at 2, 4, 6. And I just did a bitch recently at 8, just because she'd had a lot of progeny that were being bred from. But I think 2, 4, and 6 is a really good age to do your. And I do not. To me, just listening to the heart not only tells you about murmurs, it doesn't tell you about flow rate. It doesn't tell you what size the chambers are. So it's got to be done, by a veterinary cardiologist. And then I do my eyes.
So not just bloods.
My eyes on the same schedule. I do the 2, 4, 6. But since she was alive at 10, we just did Martina's eyes as well, because we were like, wow, that bitch has a lot of progeny all over the place, and I want people to know that things were tickety-boo. And we put all of that up on the OFA website. More recently with the genetic testing, so the younger dogs, where I've started doing the genetic testing for things like the centroneural myopathy, and I've also now started to put those up on the OFA website. But I would actually prefer to have somewhere like we have with Wheatons, which is a website that is centered around the Great Dane and a database with our pedigrees, with our health testing, with our DNA, with our age of death, cause of death, and in the future we may, also be able to look at things like, so Danes are very good at things like FastCat, and certain companion events, and it would be good to know in the future if we can look at the DNA and see, why certain dogs are so good at this. Is it prey drive? Is it athleticism? Is it fun? But for those of you who haven't done it, it's sort of like coursing with a plastic bag. But the dogs absolutely love it. They go tearing after that thing. And Danes can be really fast. You see their hound side coming out very nicely in it.
I'll tell my bitch.
Does that answer your question?
Now that she's very fast. She's lady snoring. Like, don't ask me to move.
Okay.
Ace is snoring.
Betty's laid there like that. I'm a hound. I'm a hunter.
I'll go get a puppy that I promised Sandy.
Please. Can I see Dory as well, please? Oh no.
She's sleeping.
Tell her that Auntie wants to give her a
She's been tear-assing round here, and now she's finally gone to sleep. I ain't waking her up.
Well,
There's the other one down there.
Tasha's been working very hard Pash is a real working dog because I think that we've got rats in the garden because of my. So he's been on.
Can you see that one, Sandy?
What? Where? Where's that?
That's Ace, flat out on the sofa. Can you see him?
Has he been a good boy?
Snoring his head off.
He's not doing much work.
Pardon?
Ace is not doing much work.
Oh, here we go.
Hang on. Let's have it. Oh, look. Bit closer, bit closer. Hello.
Oh, look at them.
Oh.
We can't see.
Yes.
I can't see. Where are you? Feet, Neil.
Oh, they're
Where's
lovely.
Neil? We can't see it. We can see
We
something.
can't see anything.
Is that the naughty one? Is that the one naughty one?
This is the naughty one.
Ah.
Oh, look.
This is the little angel.
Oh, no.
Oh, they're lovely. Absolutely gorgeous.
Mum's quite concerned that I have them. Pictured.
Is she coming to see us?
Where's
Here
Neil?
she comes.
Oh, we've got a form. The next black I have will be cornbread, without a shadow of a doubt.
Oh, good girl.
Oh,
Good
my
girl.
baby's come to join us now.
Oh,
Aw.
Okay,
Hello,
Sandy.
Ruma.
Let me go back.
okay, everybody. Thank you, Neil.
Thank
Thanks,
you.
guys.
Hang on, don't check off yet, Neil, because Neil's out of sight. We've got to thank him properly. Thanks, everybody, for attending. It's lovely
Thank you.
to see good attendance. It's fantastic. And I don't know how, I think if we could all get some sort of system going, David's got something that, I'm too old to start a database, even though
I tried to start a database some years ago and nobody wanted to know it.
the best database to copy or to have allegiance with is the Quintessa one, because that goes right through our country, Australia and America.
Yeah.
You know?
Can I just say, how can you have a database when half the breeders don't test anyway?
Well,
Did it not ought to come from the Kennel Club?
No, we can have our own database. But
Yeah,
people,
but what did you have on there? Because half the breeders don't hip score.
well, it might encourage people to do testing if they know that the dog is going to be on this board.
Could we not have Maria, could we have something from the Breed Council that, you know, because we're starting off with all our tests now, and it's knowing which tests to do. So it would be really great if we could get some guidance and maybe, you know, as a breed club, we say these are the tests that we should do.
Well, this is what we're looking into, hence the tests that David and the subcommittee we've just put out now. Hopefully the findings from that, when we've got it, we will know what we need. So we are in the process of doing this. I tried it years ago and no one wanted to know, but things have moved on. So hopefully we will, with the results of this test, we'll know what to test for and we can get it out there and get a database going, which I think is brilliant, and every breed should have one.
Sorry, I've only just walked back in, so I couldn't caught the end of that.
Yeah,
But yeah,
actually,
I agree with what
Sandy was just bringing this meeting to an end, David.
Okay.
Thank
Shall
you.
I shut up then?
But with saying what can we do? What can we do? Breed Council. Let's think about sorting out when you get the results, Dave, from your what do you call it? From your, what's the word? I'm getting old.
Survey.
Yeah, from your survey, your POG, so that we can possibly think about doing a database. I don't mind. I think I'm a bit old to be doing a database now, but you know.
Personally, I use my health test as an MOT. So you wouldn't buy a car without an MOT.
No.
You can't guarantee that car's going to be safe, but you can. When breeding, you need to sort of know that you're breeding good stuff, and the only way you can do that is by doing the tests and having these results. And with so many breeders not doing the tests because they're worried of what the results are going to be, then you're not going to go any further. It's yes, you'll still have problems, you'll still have errors, but a lot of errors and problems are you can't stop them. At the end of the day, you're messing with nature, and these things will happen whether you do these tests or not. But I believe that you should test.
But the more people get involved in testing, the more people who get involved in the survey, the more information that we have, you know, and we can make better informed decisions going forwards then, you know. So it's down to everybody to get involved, and then for the breed council to come up with a plan that works for everybody, doesn't it, and takes us forwards rather than sort of standing still or going backwards.
Okay, Dr. Neil is just showing us what are you showing us?
So this is what in the US we've got our CHIC program. It's the Canine Health Information Center. It's an offshoot of the Orthopedic Foundation. But, you know, here's up on top is the dog's information, and then all of the testing that's been done, those required for CHIC, but any additional testing is also listed. And if you remember at the Great Dane Futurity, if both parents have a CHIC number and your dog wins the Futurity, there's a special prize that's awarded to that dog because of that health information. The other thing we do is we have champions of health. So dogs that have been very successful in the breed and also have all this testing information can be given a special award, champions of the breed. So the idea I'm coming up with here is we need positive reinforcement of doing it. You know, it's not all with the stick, but we need to have lots of carrots in there. So giving recognition to the dogs that are both excellent in the conformation and the performance ring, but also have all of this health testing information is the positive way to go forward.
Well said.
And each CHIC, you know, the requirements for each CHIC are determined by the parent club in the US, because, you know, what's relevant to testing a Great Dane has nothing to do with what's relevant to testing a soft-coated Wheaten Terrier.
Okay.
Good. Right, well, I'll hand you over to Neil, our secretary, to say thank you. I'll say thank you to Dr. Neil. Again, lovely to see you. And
yeah, thank you very much
and
indeed.
Our Neil. Neil? Neil?
Which Neil are you talking to?
Neil Morgan now. I'm handing over to
No,
Neil.
Neil, I think it's been brilliant again, and I think the more and more we do this, the more we'll kind of understand it, because a lot of it's way above our pay grade, but it's starting to sink in. It really is, and I think it's the message is getting out there, so I think it's good. Yeah.
I promise I'll make that scientific mumbo jumbo just easier to work with as we go by.
Yeah, but it is definitely sinking in, because with the first one that you did, when you sort of went away and re-looked at it and analysed it to the PowerPoint presentation, and then with this one, it's become people I think are understanding it a lot more. So it's only for the good. Absolutely.
Great. Yeah. I thoroughly enjoy doing this, and I thoroughly enjoy being active in the UK scene.
Yeah.
My Great Dane heart and roots started there.
Thank you.
Lovely. Thanks, Neil.
Thank you. Have a good evening, everybody.
Bye.
Bye. Take care.
Bye. Lovely to see you all.